So here we go with the part 2 of the Genes Within Populations lecture. Again, remember what we talked about with evolution. It is the change in a population, not the individual. And I'm going to keep stressing that over and over and over because that is the biggest misconception people have. So change in a population, specifically the genetics, the genes or the alleles of the population, change over time if the population is evolving. So what we want to look at now is how do we measure this? How do we actually tell if a population is changing? What are the features or factors or things that we can look at to determine that? So what we use is a thing called Hardy-Weinberg principle. This is a way to actually measure changes in allele frequency. So is the percentage of big A versus little a changing in your population over generations? Not one generation or two, but hundreds, thousands of generations. So again, our challenge is, if I'm looking at oak trees, I can't watch them change in my lifetime. I'm not going to live long enough. But if I'm watching fruit flies, those suckers reproduce every couple weeks. If I'm watching bacteria, 20 minutes, I can see the changes. So think about new bacteria, MRSA, tuberculosis, things like that that are resistant to antibiotics. That is evolution in front of our eyes. So we can measure those changes in the allele frequency. So to help measure it, we use a thing called Hardy-Weinberg equilibrium. Now, Hardy-Weinberg says, if these factors occur, you do not have change. If these 5 factors are present, and they all have to be present, it's not if only one is there or two, it's all 5, it's a 5 or none. If all 5 of these factors are present in your population, then you do not have evolution. Okay, so first thing, no mutations are allowed or occurring. Let me change it to allowed. You cannot have a mutation in a population in order for it to be not changing. If there's mutations, changes are going to occur. Number 2, gene flow is not allowed. Let me retype the first one instead of no mutations. So mutations are not allowed. Okay, so mutations are not allowed. Population is not evolving. Gene flow is not allowed. Populations will not change. Mating between individuals, sorry about the typo here, mating between individuals is completely random. Close your eyes, mate with whoever you bump into. Has to be that way. Mating has to be completely random. Your population size is very large. And natural selection is not allowed. Okay? So all 5 of these have to be there. They all 5 have to be present in a population in order for it to be in equilibrium or evolution is not occurring. All right, so that's the key. The frequencies, the allele frequencies will stay the same if all 5 of these things are there. You can't have a mutation. You cannot have gene flow. Mating has to be completely random. Population has to be large, and natural selection is not allowed. So if all 5 of those conditions are met, populations will not change. So let's look at the reality of the situation. That's impossible. That's impossible for any length of time. So mutations randomly occur. DNA messes up. you could block some of these things. If you said, I want to have goldfish in my fish tank, and I'm going to have them live there for the next 100 generations, you could block some of these things and prevent the population from changing. But you can't realistically prevent all of these things. That's why species are always evolving. It's just a matter of how fast can we see it in our lifetimes, what are the actual changes that are visible phenotypically, what is going to be expressed, and do they create new crazy exceptions or crazy variations of the existing or the original species. Okay, so with Hardy-Weinberg, we use an equation to actually calculate the allele frequencies. And in this equation, we're looking at simple dominant recessive allele here. Black is dominant to white in our cats. Big B for black, little b for white. Now you could be homozygous black, 2 big B's, heterozygous black, big B, little b. And then white, recessive, 2 little b's. So we use the letter P to represent the dominant allele and the letter Q to represent the recessive allele. So P squared means homozygous, big B, big B. 2PQ represents your heterozygous genotype, and Q squared represents the recessive cat, the little b recessive alleles. Now, P squared plus 2PQ plus Q squared equals 1. So we need a little bit of basic math here, a little bit of algebra to solve this equation. So let's take a look at an actual population. And you can determine all this by looking at the population, looking at the phenotype, and then figuring out what the frequencies are of these alleles in the population every generation. Okay? So you start with the phenotypic frequencies. And let's say you got 84% of your cats are black, 16% white. So you have 100 cats, that's a crazy cat lady, 100 cats, 84 are black, 16 are white. So what we do is we figure out what are the genotypic frequencies with this. So in order to do that, this is your recessive individual. If it's 16% of the population is recessive, then 0.16 is going to be the genotypic frequency of those individuals. Little b, little b. From that, what we can do is extrapolate and work our equation to determine the frequencies of the various gametes that we're going to have in our population. Okay, so don't get too overworked right now. If you're looking at this and your math isn't the strongest and you're saying, I just am not getting this. This is something that's done during the evolution lab. So I wanted to make sure I introduced it, make sure you guys started to get a little bit of a feel for how you work the equation, but that's something you'll work in the evolution lab. Okay? The other thing that was not presented in the previous slide, let me put it here. If you're looking at just allele frequency, not genotype now. Here's genotype, big B, big B, big B, little b, little b, little b. Allele frequency, oh, let me reduce font size here. The allele frequency can be calculated with this equation. I'll get it, allele. Oh, come on. Oh. So to determine allele frequency, P plus Q equals 1. So let's do some quick math here. Let me move this over, give us a little room. P plus Q equals 1. In the scenario of our cats, what we have is 16% of the population as BB. That's B squared. So if you took the square root of 0.16, you would have the frequency of Q. You plug that in there. With that, you can solve the equation to determine the frequency of P. So again, you guys will do this in lab. I just wanted to introduce it now so it doesn't look so crazy and unfamiliar when you get into lab. Okay, so now that we know how to calculate it, what we want to look at are the 5 factors that lead to evolutionary change in a population. Okay, know these. Absolutely know these. Double red star means you need to know it. It's a core concept for this unit. Okay, first one, mutation. Mutations are raw genetic material. Okay, this changes the genotype. Generally, though, mutations happen in very low rates. Okay, we don't see it a lot, but it does happen. Remember, DNA replication, roughly 3 errors persist in the cell cycle. That creates differences. Now, in the case of the flower here, the white parts on the petal versus the pink might not be a big deal. But in the case of the frog, you got an extra leg, that's problematic. All right, so that is our first factor that leads to evolutionary change. Second factor, gene flow. Genes moving from one population to another. Okay, now this may or may not be present depending upon the type of species you're working with. All right, so birds, geese, Canada geese migrate. Oh, wrong one. Canada geese migrate every year. They're moving around. They're spreading their genes. Coconuts float hundreds or thousands of miles in the ocean, moving their genetics. Bees pollinating one flower to the next, to the next, to the next. That's all gene flow. Some populations are isolated, though. They're stuck in a small section of a mountain range. They're at a certain elevation, and they can't reproduce with another population because the mountain range is very limited. Sometimes gene flow is there, other times it's not. Okay, so that's our second variable that contributes to natural selection, or contributes to evolution. Third variable, non-random mating. All right, so unfortunately, Brad and Jolie, probably should take them out and put a different picture in there. But you think about non-random mating. It's also known as sexual selection. And this only applies, obviously, to species that reproduce sexually. But likes tend to mate with likes. All right, so guys, probably not going to have a chance with Angelina. Ladies, probably not a chance with Brad. Likes tend to mate with likes. Attractive, rich people tend to marry and reproduce with attractive, rich people. Just the way of the world. Peacocks. You got this male here with this big, bright, showy flower, or showy feathers, a big tail fan. He's going to appeal to the strongest, healthiest, most attractive female in the flock. So you can block this if you limited how many peacocks were there and forced matings, but that's not realistic. That's not nature. That doesn't happen in a natural setting. Okay, so our third factor that contributes to it. The fourth one is something called genetic drift. Now, genetic drift is really just pure chance that the allele frequency changes. Just blind luck, pure chance. All right, so what we want to look at are the 2 examples of genetic drift. One is called the bottleneck, and the second one is known as the founder effect. We will look at both of these in the next